Good day, I'm Jessie Nisiakan, and here are the start of our report, which is Chapter 9, Gene Therapy. So, the body composed of trillions of cells. Cells are the basic building blocks of all living things. The common center of each cell is called nucleus, and it's contain chromosomes. So chromosomes are made of DNA, which is the body's hereditary material. So its genes is typically present in two copies, one each from parents, and protein, which are molecules that helps body grow, regulate, and sustain itself, are made by the body according to instructions found in portions of DNA called genes. So, about 20,000 genes made of an individual, the majority of which are universal. Each individual has a small number of slightly unique genes, though people differ in their characteristic due to this little genetic variation. So there is a probability that the DNA sequence of genes could occasionally have a replacement, deletion, or duplication. This is referred to as such a mutation, and it may result in absence of improper function of a required protein. A mutation might be inherited from one parent or acquired later in life. Genes modification can affect our health, whereas some are unharmful. Genetic illnesses can occur from genetic or genes mutation. So let's move on to gene therapy. So gene therapy is a technique that modifies a person's genes to treat or cure disease. So it is a technique that use materials, so a piece of DNA for a long-term treatment or genetic disorder. So gene therapy has provided treatment for a curable disease that previously had only temporary remedies. So some of these diseases are partly or fully caused by genetic mutations. So such as cardiovascular disease, neurodegenerative disease or disorder such as Parkinson disease, vision disorder, blood clotting disorder such as hemophilia and diabetes.
Good day everyone. My name is Kay Sergio A. Orquesa from BPED 40. And now, I'm going to discuss about approaches of gene therapy. Gene therapy works by replacing or inactivating disease-causing genes. In some cases, gene therapy introduces new genes into the body to treat a specific disease. With gene therapy, doctors deliver a healthy copy of gene to cells inside the body, and this healthy gene may replace a damaged or mutated gene. Inactivate a mutated gene or introduce an entirely new gene. Carriers, called vectors, transport these healthy genes into cells. In most cases, the vectors are modified viruses that do not cause disease. Vectors may also be certain types of bacteria or circular DNA molecules or plasmid DNA. Additional methods to package and deliver genetic material are also being actively investigated, such as the use of nanoparticles and encapsulating lipid molecules and the use of electric currents. Injection or intravenous or IV infusion introduces vectors into the body. In some cases, doctors collect cells from a patient and vectors in a laboratory and return the vector containing cells to the patient's body through injection or IV infusion.
For continuation of our report, let's move on to the types of gene therapy. First is somatic cell gene therapy. Therapeutic genes transfer into the somatic cells. For example, introduction of genes into bone marrow cells, blood cells, skin cells, and etc. will not be inherited later generation, and at the present all research is directed to correct genetic defects in somatic cells. And in the second is germline gene therapy. Therapeutic genes transfer into the germ cells. For example, genes introduced into the eggs and sperms. It is heritable and passed on to the later generation. And for the safety, ethical, and technical reason, it is not being attempted at present. And I am Janina Paula S. Balino.
Good day everyone, my name is Rona May de Abrea and I am here to discuss to you the gene therapy strategies. First, let's talk about gene augmentation therapy or GAT. In GAT, simple addition of functional alleles is used to treat inherited disorders caused by genetic deficiency of a gene product. Example, GAT has been applied to autosomal recessive disorders. So, dominantly inherited disorders are much less amenable to GAT. So, gene therapy strategies or GAT consists on the delivery of a new protein coding gene to treat a disease. Gene replacement therapy is directed at monogenic recessive diseases that are caused by non-functional genes consisting in the delivery of a non-mutated mutated form of the affected gene to treat the disease. Next, targeted of killing specific cell. It involves utilizing genes encoding toxic compounds or suicide genes or prodrugs, agents in which confer sensitivity to subsequent treatment with the drug, or to kill the transpected or transformed cell. So, this general approach is popular in cancer gene therapies. So, killing of specific cells is aimed to insert DNA into a disease cell that causes that cell to die. Next, targeted inhibitions of gene expression. So, this is to block any disease gene or a new gene expressing a protein which is harmful for a cell, and this is suitable for treating infectious diseases and some cancer.
Methods of gene therapy. In order to treat a sickness or aid the body in fighting disease, gene therapy research look to see if a new gene may be utilized to replace or inactive a mutant gene. A transporter is usually employed to convey a gene into a cell, and a vector is a type of transporter. A mutated virus is used to create a vector. The virus viral genes are eliminated before to use as a vector. Once the virus has been altered, its goal is to deliver the desired gene to the cell without causing illness. Once inside, the desired gene should allow the protein to resume its original function. Vectors can be injected into a particular body tissue or supplied intravenously, which means they are administered into a vein. The delivery of vectors to a particular bodily region can also be accomplished by other processes, such surgery. And there are two mainly approaches for the transfer of genes in gene therapy. So the first one is in vivo therapy, transfer of genes directly to cells inside the body. A vector usually carries the new genetic material into the body through an injection in blood or directly to the target organ. And the last one is ex vivo therapy, transfer of genes into patient cells outside the body. Once the newly altered cells are prepared, they're delivered back to you, usually by an intravenous infusion.
Good day everyone, I'm Ang Lester Valdez. Ex vivo gene therapy. It involves the genetic modification of cells outside of the body to produce therapeutic factors and their plantation back into the patients. In this mode of gene therapy, genes are transferred to the cell grown in culture. Transformed cells are selected, multiply, and then introduced to the patient. The use of autologous cells avoid immune system rejection of the introduced cells. This approach can be applied to the tissue like hematopoietic cells and skin cells, which can be removed from the body, genetically corrected outside the body, and reintroduced into the patient body where they become engrafted and survive for a long time period of time. It is a medical procedure in which an organ, cells, or tissue are taken from a living body for a treatment or procedure, and then returned to the living body. In vivo gene therapy. In vivo method of gene transfer involve the transfer of clones genes directly into the tissue of the patient. This is done in the case of tissue whose individual cells cannot be cultured into vitro in sufficient numbers like brain cells, or where implantation of the cultured cells in the patient is not efficient. In vivo gene therapy uses viruses or methods to deliver genes directly into your cell. Its simplicity and reduce the volume of materials introduced into the brain. The efficiency of gene transfer and expression determine the success of this approach because of the lack of any way for selection and amplification of the cells, which take up and express the foreign gene. Ex vivo gene therapy takes cells from your body, modifies in lab, and puts them back into your body, while in vivo gene therapy, new genes inserted directly into your body.
Good day, I am Blessy Bondok and I'm going to continue the discussion about gene therapy. Target sites for gene therapy. So sabi dito, therapeutic genes have to be delivered to specific target sites for a specific type of disease. So hindi lang pala basta-basta na di-deliver yung therapeutic gene. May specific target sites pala for a specific disease. This table describes the list of such disease and their target sites for gene therapy. So, let's discuss first the first disease in the table, which is cancer. So, cancer is a disease na cause ng uncontrolled division of abnormal cells in a part of the body. Makikita din natin sa table ang mga target cells for gene transfer. For gene transfer in a cancer, these are the target cells: tumor cells, antigen-presenting cells, blood progenitor cells, T cells, fibroblasts, and muscle cells. So, ano nga pala yung tinatawag nating gene transfer? Gene transfer is the introduction of foreign genes or gene sequences in a target cell's population using non-viral or viral vectors to treat a particular disorder in affected individual for sustained expression of a transgene cassette versus application of the therapeutic product itself that exhibits a short pharmacological half-life. Next is inherited monogenic disease. So, ano nga ba ang sakit na ito? So, ito pala are caused by mutation or alteration in the DNA sequence of a single gene. They are known as Mendelian hereditary disease as they are transmitted to offspring according to Mendel's law. At ang mga target cells nito for gene transfer are lung epithelial cells, macrophages, T cells, blood progenitor cells, hepatocytes, and muscle cells. So next is infectious disease. Infectious disease are disorders caused by organisms such as bacteria, viruses, fungi, or parasites. Many organisms live in our body. They are normally harmless or even helpful, but under certain conditions, some organisms may cause disease. So, T cells, blood progenitor cells, antigen-presenting cells, and muscle cells are the target cells. So next is cardiovascular disease. Cardiovascular disease is a group of disease affecting your heart and blood vessels. This disease can affect one or many parts of your heart and or blood vessels. A person may be symptomatic or physically experiencing the disease or asymptomatic, not feeling anything at all. And the target cells for gene transfer in this disease are endothelial cells and muscle cells. So next is rheumatoid arthritis. Rheumatoid arthritis is a chronic inflammatory disorder that can affect more than just your joints. In some people, the condition can damage a wide variety of the body systems, including the skin, eyes, lungs, heart, and blood vessels. Rheumatoid arthritis occurs when your immune system mistakenly attacks your own body's tissue. For gene transfer, the target cells in this disease is the synovial lining cells. So last is cubital tunnel syndrome. Cubital tunnel syndrome is a problem with the ulnar nerve which passes through the inside of the elbow. It causes pain that feels a lot like the pain you feel when you hit the funny bone in your elbow. Cubital tunnel syndrome may happen when a person frequently bends the elbows, lean on their elbow a lot, or has an injury to the area. The target cells for gene transfer in this disease is nerve cells.
Good day everyone, I am Justin D. Catle from BPED 4D. And I am the one who will discuss the advantages and disadvantages of gene therapy. First is gene silencing. In the case of HIV-infected person, gene therapy and gene silencing can protect the patient from pain and suffering before the disease progresses. So, the advantage of gene silencing is kaya niyang protektahan or kaya niyang iiwas yung pasyente sa paghihirap or sa dulot na sakit ng disease bago magpatuloy yung sakit niya. For example, isa kang HIV infected person. Pag nag-take ka ng gene therapy, meron silang gene silencing na tinatawag. And dito, may iwasan na makaramdam ka ng sakit or ng paghihirap bago mag-progress yung sakit mo. Next is, gene therapy is potentially used to eliminate and prevent hereditary diseases such as cystic fibrosis. It is also a potential way to treat heart disease, AIDS, and cancer. So, hindi lang pala pinoprotektahan ng gene therapy ang pasyente, kung hindi, paraan ito para maiwasan na maisalin sa susunod na generation or sa anak mo, para maiwasang mamana na magiging anak mo yung sakit na meron ka. And ito rin yung posibilidad na paraan para makagamot ng heart disease, pati AIDS and cancer. So malaking tulong yung gene therapy. And now let's proceed to disadvantages of gene therapy. Disadvantages of gene therapy. First, the novelty of gene therapy method is one advantage. So, meron talagang advantage yung gene therapy. Next is, stimulation of immune system. Nakapaloob ito sa disadvantage ng gene therapy. The gene injected by virus may cause immune responses due to presence of the virus inside the body. And the pathogenic potential of viral vectors. In one case, the viral vector could improve its ability to cause illness. So, yung disadvantage pala ng gene therapy, pag nag-take ka ng gene therapy, is yung stimulation of immune system. Makaka-apekto to sa iyong immune system, and malaki din yung possibility na ma-improve or tumaas yung posibilidad na magkaroon ka ng sakit or ng illness. Dahil nga sa stimulation of immune system. And lastly, generation of genetic disorders. due to presence of multi-gene. The genetic material transferred may not necessarily enter the target cell. If it does, it may not be able at the appropriate place in the game. So, yung pinaka-generation of genetic disorder due to presence of multi-gene, isa din itong disadvantage kung gagawin mo yung gene therapy. Hindi naman pag sinabing gene therapy is papasok na agad yung pinaka-genetic material na gusto mo or yung pinaka-gamot sa gustong pasukan na to na cell or yung gustong magamot. Kung pumasok man ito sa target cell, hindi rin appropriate or hindi rin siguro mailulugar ng pinakagamot yung sarili niya sa loob ng katawan or ng target cell. Thank you.